We looked at how chromosomes move and are divided up during mitosis and meiosis. In mitosis, you'll recall, the point was to get copies of all the chromosomes into the daughter cells, thus ensuring continuity of genetic information during normal cell division. Meiosis, on the other hand, is a special kind of cell division that creates haploid cells, dividing up homologous pairs of chromosomes to create gametes used for sexual reproduction. Now, meiosis is especially interesting to us because the way that the different members of homologous pairs separate independently creates an enormous amount of genetic variation, as I said. The behavior of chromosomes during meiosis actually does more than just tell us that genetic variation is created. It actually predicts how suites of traits are going to be inherited by offspring from their parents, which is the first step we need to take towards understanding how traits are generated across generations, how traits are transmitted across generations, and ultimately how traits in a group of organisms may change over evolutionary time. It turns out that the discovery of the fundamental mechanisms by which traits are transmitted from parents to offspring actually preceded the discovery of chromosomes by many years, and was made by a very unlikely person, an Augustinian monk named Gregor Mendel, who actually unlocked the secrets of modern genetics by growing plants, pea plants, in the Abbey garden. In today's lecture, we're going to begin to take a look at Mendel's work, and we'll continue this in the next lecture, to provide a foundation for understanding how traits are transmitted across generations, an area that we now call Mendelian genetics in his honor. Mendel lived in the mid-1800s, and he began his life on a small farm in Austria. Thus, Mendel grew up with kind of an intuitive understanding of plant and animal breeding. But Mendel was drawn to theology, not farming, and so he joined a monastery. Mendel was no ordinary monk. Actually, in the early 1850s, his order sent him to study at the University of Vienna, one of the leading universities in Europe at the time, and he was taught by many of the outstanding scientists there. Most notably, for example, the physicist Christian Doppler, for whom the physical Doppler effect is named. When Mendel returned to the monastery, he began to study heredity in garden peas, and especially the effects of crossbreeding varieties of peas that had different traits. Now, at the time of Mendel's work, chromosomes, as I said, hadn't even been described, let alone mitosis and meiosis, and nothing was known about the physical basis of inheritance. But it was obvious, as it had been for many thousands of years, that offspring do tend to resemble their parents. It also was obvious, however, that different combinations of traits in parents would appear to be mixed in different ways in their offspring, sometimes surprisingly so. So scientists in Mendel's day were interested in the question of how traits could be transmitted from parents to offspring, for example, when they looked at how different kinds of plants bred or farm animals bred. Now, at that time, in the mid-19th century, the dominant theory about how inheritance worked suggested that some material from the two parents was blended together when offspring were produced. You can think of this like mixing paint. Imagine that the genetic material for a particular trait is like paint in a bucket. What scientists thought was that when offspring were produced, paint from the two parental buckets would just simply be poured together into the offspring's bucket. And the result would be some mixture of the two. So, for example, if an animal with light colored fur mated with an animal with dark colored fur, the result you would guess is some intermediate color of fur. And indeed, sometimes that's what was observed. But the blending hypothesis actually didn't account for many other observations made by plant and animal breeders. Specifically, when you think about it, the blending hypothesis predicts that over time, all variation within a species should vanish. Again, think of blending buckets of paint. Let's imagine we start out with a whole bunch of colors in different buckets, and as those buckets are, and as offspring are produced, those buckets are just mixed together. Over time, you'd imagine we just have buckets with one kind of paint, all the same color, a mixture, an even mixture of what we started out with. In the real world, however, plant and animal breeders realized that even when individuals in a population mated randomly for a very long period of time, there always remained variation among individuals. Also, and surprisingly, sometimes traits would appear in offspring that weren't present, obviously, in either of the parents. So this was the kind of problem Mendel was trying to address when he began his work on pea plants. Now, to understand Mendel's work, what I want to do is to begin with the experiment he first did, a very famous experiment that we call a monohybrid cross, for reasons that will become apparent in just a moment. Now, Mendel worked with garden peas, as I said, and it turns out that garden peas were excellent subjects for him to study for a couple of reasons. First, he could carefully control which pea plant mated with which other pea plant. Ordinarily, these garden peas self-fertilize. What that means is that a male gamete from one plant will actually fertilize a female gamete from the same plant. Many flowering plants do this. But what Mendel could do is clip off the male reproductive part of flowers before those flowers had matured, and in that way prevent the plant from self-fertilizing, and then he could fertilize that plant with his choice of pollen from any other plant. So he could control completely the genetic crosses he was doing, which of course was very necessary for his work. The second and actually more important reason that peas were a good species to study is because they display a number of inherited characteristics that occur in only one of two very different discrete forms. For example, Mendel's most famous experiment involved looking at the inheritance of flower colors. In these pea plants, flower colors occur in only one of two forms. Flowers can be purple or they can be white. It turns out that we call this kind of character a dichotomous character. It's one thing or the other. And pea plants had many such dichotomous characters, including not only flower color, but also seed color, the shape of the seed, the height of the plant. Individuals would be in one form or the other. Now, Mendel understood that the existence of these kinds of dichotomous traits couldn't be reconciled easily with the blending hypothesis, so he decided to investigate how these traits were transmitted across generations. Now, Mendel began his first experiment with parents that he crossed that came from two what we call true breeding lines. These were self-fertilizing lines of pea plants that would produce only one form of the trait or the other. For example, he would take a plant from a self-breeding, self-fertilizing line that would only produce white flowers, and another plant from a self-fertilizing line, a true breeding line, that would only ever produce purple flowers. What he then did was ask what happens if you cross a purple-flowered plant with a white-flowered plant. This is called a monohybrid cross because what he's doing is hybridizing two varieties that differ in only a single character, in this case flower color. Now, when Mendel performed this cross, he found a very surprising result. Remember, all the parents were either purple, one parent was purple and one parent was white. When he crossed them, all of the offspring were purple. Let me give you a little terminology. We started with what we'll call a parental generation, the purple and white plants, and what we're looking at now, the offspring of this generation, we're going to call the first filial generation. That's the technical term. We often refer to it as just the F1 generation. This is the first generation after the initial parents are crossed. Now, the result that Mendel got in this F1 generation of all purple plants was surprising because it completely contradicted the blending hypothesis. The blending hypothesis would have suggested we'd get an intermediate color, say sort of a light purple. But no, all of the offspring were purple-flowered as their purple-flowered parent. And the white flower color had been completely lost, or it seemed to be. Now, an even more surprising observation that Mendel made was what happened when he crossed these F1 individuals, the offspring that he got from that first cross, crossed two of those F1 individuals, all of whom had purple flowers, and asked what happened in the next generation, which we call the second filial generation, or the F2 generation. This was more surprising because what he found in the second generation, the second filial generation, was that the white flower color reemerged. He would have some purple flower colored individuals and some white flower colored individuals. Furthermore, and this was the kicker, these individuals would always occur in approximately the same ratio. In this case, there would always be about 3 purple individuals, let's just say purple individuals, and 3 and one white individual. Let me repeat that. In the F2 generation, you would find 3 purple plants for every 1 white plant. So there are 2 surprising things here. One is the white came back, and 2, the white and the purple always occurred in this same unique ratio. Now, Mendel did this kind of cross with a number of other dichotomous traits like seed color or seed shape and so forth, and he always got the same result. One trait would disappear in the F1, and it would reappear in the F2 in the ratio of 3 to 1. What could account for this pattern of inheritance? Specifically, how could you have one trait lost completely and then reappear, and always in this same proportion? In puzzling the answer to this out, Mendel essentially developed a hypothesis that explains what genes do on chromosomes when they're transmitted, even though he had no idea what genes were or what chromosomes were. And in so doing, he essentially established modern genetics. Let's look at what Mendel argued. We can divide his hypothesis into 4 related ideas. First, Mendel argued that the different versions of what he called heritable factors, that different versions of what he called heritable factors must be responsible for producing the different traits for a particular character. In other words, he said there must be some heritable factor, or he might have called it a heritable particle, responsible for purple flowers, and a different one responsible for white flowers. Now, what Mendel called heritable factors then are what we now call genes. And the different versions of the genes are what we call alleles. This is a new term. So every gene, in Mendel's case, every trait had actually, every character, I should say, had 2 different traits which would correspond to one gene having 2 different versions that we call alleles. So Mendel's first contribution then was to explicitly suggest a particulate theory of inheritance and suggest that these different kinds of particles, alleles, are passed from parents to their offspring. Again, in contradiction to the blending theory. Mendel's second idea was to conclude that each individual had not 1 but 2 different particles for each character. In other words, we would say it had 2 alleles for each kind of gene. Again, Mendel made this conclusion knowing nothing about chromosomes, but given our discussion of cell division from last time, you can see how each offspring, in fact, does have 2 copies of every gene. One from its mother, one from its father, located on the homologs of a homologous chromosome pair. Now, Mendel's third suggestion was that one allele of the 2 that an individual possessed may actually be dominant over the other allele. In other words, there could be one kind of trait, one allele, that is expressed exclusively, even if the other allele is present. We say that one allele would be dominant, and the allele that is not expressed we call recessive. Now, let me clarify this idea by going back to what Mendel actually saw in his cross in the F1 generation. Remember, he started with true breeding lines of purple and white flowers. He argued that these true breeding lines, because they always only produced one or the other flower color, must always have 2 of the same kind of allele. The purple flowered plants would always have 2 purple alleles, and the white flowered plants would always have 2 white alleles. When Mendel argued that the offspring of this cross must receive 1 allele from each parent, and thus must have 1 purple allele from one parent and 1 white allele from the other parent. Because all of the offspring in the F1 generation had purple flowers, Mendel argued that the purple form must be dominant over the recessive white form. Now, Mendel's fourth idea, and in a sense this was his most critical idea, was to argue that when a parent produces gametes, when a parent itself produces gametes in preparation for sexual reproduction, each gamete only gets 1 of the 2 alleles, the 2 copies that that parent possesses for a particular gene. Now let's think about this. When a parent then, if we're thinking about just one gene, say for flower color, when a parent, if a parent possesses the two of the same copies of the allele, 2 of the same kinds of the allele for that trait, when it produces gametes, all of the gametes will have just that kind of allele because it has no other alleles to contribute. So for example, purple flowered colored plants from Mendel's parental true breeding line would only make gametes that had purple flower colored alleles. And the same would be true of the parental white flower color alleles. The parental white flower colored, the production of gametes by the, you can see that talking about Mendelian genetics is very good practice for tongue twisters. What I meant to say is that the production of gametes by the true breeding white flower colored parent would only produce gametes with white alleles. But here's the kicker. When we have individuals that have two different kinds of alleles, for example, the individuals that were created in the F1 generation of Mendel's first cross, then they can produce two different kinds of alleles. Because they have both a purple allele and a white allele, then that you can imagine that they can produce gametes having either purple or white. And in fact, they do so in a 50-50 ratio. It's equally likely that their gametes will have one or the other allele. Okay, now let's go back to Mendel's monohybrid cross in light of what we've just been talking about. What I want to do is to walk you through a formal way of looking at this genetic cross. And in fact, you might even want to keep notes on a piece of paper as you follow along to help you keep track of how alleles are passed from parents to offspring. Now, as I do so, I'm going to introduce a little bit more genetics terminology that was developed after Mendel's day, terms he would never use, but terms that will help us keep track of what's going on. The original parental plants Mendel started with were true breeding, as I said, so they must have had two alleles for the same color. We call individuals that possess two of the same kind allele homozygous. Now, the term homozygous is derived from the idea that the zygote that would be produced, the diploid individual that is produced, has two of the same, has a homogeneous set of alleles. So if we have an individual, like our original parental plants, that have two of the same kind of alleles, we refer to them as homozygous. By contrast, when an individual possesses two different kinds of alleles for the same gene, we say that it's heterozygous. So the individuals in the F1 generation of Mendel's cross must have been heterozygous. Now, just for the sake of keeping track of things, we really need to kind of give some labels to these alleles as we move through this example. So let's say that we're going to represent the purple flower color allele with a capital letter P. And we'll represent the white flower color allele with a lowercase letter P. Now, using the uppercase and lowercase versions of the same letter is one common way that geneticists designate different alleles for a gene. You'll see in a moment why we use the letter P for this flower color trait, and specifically why the capital P refers to the purple allele. By the way, because it's easier to say, I may sometimes refer to a capital P as a big P and a lowercase p as a little p. Now, as I said, because the original parents in Mendel's cross were homozygous, they could only produce one kind of gamete. And we've already talked about now why it makes sense then that their offspring should all have purple flowers. If one of the alleles is dominant to the other, and specifically if the purple allele is dominant to the white allele, these F1 offspring all have one big P and one little p. And because they have a big P, which is dominant, they'll all be purple, regardless of the fact that they have the little p white allele. Now, that's why we use big P and little p in this case, right? Because in this case, the purple flowered color trait is the dominant trait. So we call the trait, we label the trait by the name of the version of the trait that's dominant. And in Mendelian genetics, we would use a capital letter to designate the dominant form and the small letter to designate the recessive form. Okay, at this point, I need to introduce a couple of other important terms. And these terms are what we call the phenotype and the genotype of an individual. Now, an organism's phenotype refers to what it looks like. It refers to the traits that that organism expresses. So for example, we would say of a purple flower-colored plant that it had the purple phenotype. The genotype of an organism represents its genetic makeup. Now, the genetic makeup of an organism clearly will have something to do with the traits that the organism expresses. In other words, the genotype of an organism clearly will have something to do with its phenotype, but it's important to keep in mind that the genotype and phenotype of an organism may be related in more complex ways. Specifically, a given phenotype might be produced by more than one genotype. Let me explain this by going back to our pea plants. Because the purple allele is dominant over the white allele, there are two possible genotypes that could give rise to the purple flowered phenotype. Individuals having two big P alleles, two purple alleles, clearly would have the purple phenotype. But so would individuals having one purple allele and one white allele. In other words, one big P and one little p. Both the big P and the big P little p genotypes yield the purple flowered colored phenotype. And that's because the purple allele is dominant over the white allele. Now, on the other hand, the only way we can get the white flower colored phenotype is if we have a genotype that has both recessive white flower colored alleles, in other words, little p, little p. This helps us explain the surprising findings from Mendel's first cross. You see, the white flower colored phenotype had disappeared completely in those F1 individuals, but not the white allele, which was hidden in the heterozygous genotypes of some of those individuals, the ones that had one purple and one white allele. Now, let's look at what Mendel found when he crossed these heterozygous F1 individuals together again. Remember, when he crossed the F1 generation, which was all purple phenotype, and had both, and was all the heterozygous big P, little p genotype, he found that the offspring in the F2 generation produced both purple and white phenotypes in the ratio 3:1. Now, to understand that 3:1 ratio, let's consider again the kinds of gametes and the proportions of gametes that those F1 individuals could produce. Because these individuals in the F1 were all heterozygous, as I said, they could produce two kinds of gametes, either a purple allele containing gamete or a white allele containing gamete. In fact, each individual will produce equal numbers of both types of gametes. This makes some intuitive sense. I mean, forming gametes, if we're just talking about one gene with two alleles, is a little bit like flipping coins. We've got two possibilities. We can come up with a heads or we can come up with a tails. So if an individual has two possible alleles that could contribute to gametes, you can imagine that half the time the gamete will come up with one type of allele, and the other half of the time it would come up with the other kind of allele. Now, the key question to ask is what proportion of individuals in the F2 generation will have the possible genotypes that could be formed by joining the gametes that the F1 individuals produce. Now, there are a number of ways we could work this out. We could work this out with more probability theory, talking about the ways we would combine coin tosses. But an easier sort of shorthand way to think about this is to use a convention that we call a Punnett square. This was named after R.C. Punnett, one of the famous geneticists of the early 20th century. Now, exactly how this method works isn't important, but let me see if I can describe it to you to help you understand the results of Mendel's F2 cross. Now, the way you get a Punnett square is first to draw a table, or you might think of this as a grid, with 2 rows and 2 columns. Okay, so this table or grid has 4 cells in it. Next, what we do is we label both the rows and the columns of this table, each with the 2 possible gametes that could be produced by F1 individuals. In other words, along the top of this grid, we'll have one column that has big P on top of it and one column that has little p on top of it. Along the side of this grid, we'll have one row that has big P alongside of it and one row that has little p alongside of it. Now, the point of making this grid is that it helps us think about how these different gametes can combine to form diploid genotypes in the offspring when these gametes are joined. And if you fill out the grid then by connecting the two alleles possible for each cell, you see that you get 3 possible genotypes. This isn't very surprising. We can get big P individuals, we can get some big P individuals, and we can get some individuals that have both the little p recessive alleles. But what you also see is that of the 4 cells in this table, 1 cell will have the homozygous dominant genotype, big P, 1 cell will have the homozygous recessive genotype, little p, but 2 cells will be represented by the heterozygous genotype. And what this represents is the fact that there are 2 ways that an individual can inherit the 2 different alleles. It could get one of the dominant allele from either its mother or its father. The point is that this would suggest that in the F2 generation, we have the 3 genotypes, but they occur in a ratio of 1 to 2 to 1. 1 homozygous dominant to 2 heterozygous individuals to 1 homozygous recessive individual. Now, Mendel didn't know how to measure those genotypes directly, remember. He could only look at phenotypes. But those heterozygous genotypes, the big P, little p, all had the dominant phenotype. So if you ask what are the phenotypes of the genotypes produced in the F2, you see that you would expect there to be 3 purple individuals for every 1 white individual. Because only one of those cells in our Punnett square, the one that has the homozygous recessive allele, will give the recessive phenotype. And the other 3 cells will all yield the dominant phenotype. So by looking at this Punnett square and understanding that we have to look at the genotypes underlying the phenotypes, we can explain why Mendel found and always found a 3 to 1 ratio of phenotypes in his F2. It's because that 3 to 1 ratio of phenotypes represented a 1 to 2 to 1 ratio of the 3 possible genotypes. Now, Mendel's brilliant insight was to be able to put all of this together without knowing anything about genes or chromosomes or what the physical basis of all of this was. He just figured this had to be the way it was. He realized that the loss of the recessive trait in the F1 generation was because that recessive trait was no longer expressed given the presence of the dominant allele in heterozygotes. And then he inferred that if heterozygous individuals mated, they must have this 1 to 2 to 1 ratio of his heritable factors, which given the dominance of the dominant allele would yield the 3 to 1 ratio of the phenotypes he observed. We now refer to this key idea of the existence of 2 different alleles in each individual, and the way that these alleles will separate into cells and then recombine in sexual reproduction. We refer to this idea as Mendel's law of segregation. Now, Mendel's law of segregation is significant for a number of reasons. First, it completely refuted the older blending hypothesis for trait transmission and demonstrated that heritable factors must be particulate, basically laying the idea for what we now call genes today. Second, what Mendel's work did was to provide us now with a framework for looking at more complicated patterns of how traits are transmitted between parents and offspring, how sets of traits could be transmitted between parents of offspring, which allowed us a more complete understanding of the genetic basis of inheritance. And we'll pick up next time by looking at the next kind of cross Mendel did, which allowed him to more fully develop his ideas.